Так, can you try one more time?
Oh yes, I think there is a success.
Yeah, we can see it.
Can you see it?
Yep.
Can you see the name of the presentation, right?
Right. You can start.
Okay. Great, thank you. Hello everyone, my name is Nina Nikitina. I'm a biomedical engineering graduate student from Boise State University. And I'm originally actually from Kazakhstan, born in Zelenograd and raised in Astana. So it's a real privilege for me to speak with you today. And I'm excited to share my research journey, including how and why I developed a computer program to reconstruct complex filamentous-like structure in cells. Let me start by telling you a little bit about the lab where actually I do my research. In the mechanical adaptations laboratory, we study how aging, exercise, and unloading conditions like bed rest affect stem cell behavior and function. Our main funding comes from the National Institutes of Health, specifically the National Institute of Aging. However, we also get support from NASA that might sound surprising, but spaceflight, where there is no gravity, is a perfect way to study unloading effects. Astronauts often face bone problems like osteoporosis due to the lack of gravity. And let me just move it here. Now, many of these conditions, they affect how tissue responds to mechanical forces. And the key player in this process, can you see my pointer?
Yes. Yep.
Great, so it's the cell, the mesenchymal stem cells to be precise. And from now I will refer the cell just as stem cell for the rest of the presentation. And these cells are known for the ability to sense mechanical changes and adapt by becoming different types of tissue cells. So let's take a closer look to these fascinating cells. In this beautiful microscope picture, you can see the stem cell. The blue oval in the center is the nucleus, and the green filament structure, the outline, the shape of cell is actin fibers. And every time when I refer actin in this presentation, I mean actin filaments, also referred as fibers. So when I say actin, fiber, or filament, I refer to the same thing, the colored green structure on that particular image. And these actin fibers, they are essential for cell to thrive. They determine cell shape, allow cell to move. And what is more important for my research, actin fibers, they are like a sturdy bridge, connect our cells, nucleus, the common center where DNA govern protein production, to the dynamic environment. In this lifestyle video, which I actually took in our lab, you can have a peek on how fibers interact with nucleus. It's just 10 minutes, but you can see there are a lot of movement for so small cell, and the fibers direct movement of nucleus and affect its shape. And this connection between actin and nucleus allows the cell to mechanically feel what is happening outside. When a person exercises, for example, and then adjust to the current situation, like producing proteins, which are building block of cell, which are required in this particular moment. So not surprising why our lab was interested to reconstruct this fiber structure specifically within nucleus area. And so having this reconstruction, we can compare actin architecture between different conditions like control and treatment cells, or we even can model fiber behavior and nucleus response. This slide shows the process of creating a 3D image of cell. So we're stacking just multiple 2D images together, like taken from the top to the bottom of the cell. And typically I take from 30 to 40 slices to capture the whole cell on resolution, which I showed you earlier. Now, when we have the 3D image, we want, let's say, to count number of filaments, measure length and volume of each in 3D, right? But intricate pattern makes the task actually very challenging. And it took 4 hours for researcher to make these measurements, even with help of semi-automated tools. But 4 hours, it's not very long time, it's pretty doable. However, our last experiment required analysis of 200 images, which convert to unrealistic 800 hours. And this is where my research comes in. What I did, I created a fully automated program that reconstructs 3D structure of actin fibers, and it takes as an input this 3D image. As an output, it produces the structure with all the measurements which are necessary. This is important to remember that microscope images that we use for reconstruction, they are actually 3D. Reconstructing these overlapping fibers is a real challenging, but when we are looking from the top. So we thought, why not to change perspective and look at the cell from the side? What I mean by this, look at this beautiful green fiber. And if we slice cell cross-sectionally, on each cross-section it became just small red dot. Then what we need to do is just connect the dots together to recreate the fiber full volume. So we developed an image processing algorithm that does this for us. The algorithm consists of 4 parts: pre-processing, segmentation, reconstruction, and visualization. As an input, we have 3D microscope image here, and start our pre-processing. Here we apply a couple of standard computer vision techniques to improve image quality in general. Also, we rotate the image, so the majority of the fibers will be aligned with one axis, and just for processing convenience. And finally, the most interesting step is splitting structure in multiple cross-section images here on the right. Instead of looking from the top, now we look from the side, and we see nucleus. This is on the right, and nuclei-associated actin fibers, this image, and you can see these dots, right, which we've seen earlier. And now we have roughly 500 this cross-sectional view images for actin fiber and 500 for nucleus as well, for one cell. So this 1000 image is input for our next step. Now I want to segment these cross-sectional images and label each pixel image where white is nucleus, in this particular example, the object of my interest, and black is the background. As I mentioned, we have roughly 500 for nucleus and 5 for actin. For single cell, well, I can manually label maybe a couple of images, but labeling thousands, it's a very time-consuming task. So I taught computer to do it for me. For this task, I'm using the famous UNet neural network, which was initially developed for analyzing medical images. And actually, I'm using 2 different networks. One was trained on cross-sectional images of nuclei in the previous slide, and another for actin. The input to the model is my cross-sectional images, and the output is a binary masks showing the location of actin fibers on this example. Now we're ready for reconstruction when we have all of this labeled mask. So we have all our segments, and to reconstruct actin, we'll look at cross-section image layer by layer. So we want to connect these dots on a different layer to reconstruct one fiber, and we use just 3 simple rules. If we see dot, On the same place, in two consecutive layers, like in this yellow one, we just grow fiber. If one dot overlap with two dots on the previous layer, we just decide on the bigger overlap which fiber to grow. And if there is no overlap, we just starting a new fiber. Finally, we would like to visualize result, and this is my favorite slide because here you can see the real image on the top, and you can see reconstructed results. So we have each color actin is separate instance, and we can get all the data for each actin. Again, I input a 3D imaging algorithm, and the rest of data is depicting the structure of fiber and results as an output data here. So now this task takes just 20 minutes. As just a reminder, it was 4 hours before. And what is the best part? This program can be run in parallel on cluster. So we can theoretically analyze 200 images in 20 minutes too. And this data will be has high precision, and they will be reproducible as well. In reality, I do it maybe in a day, which is not bad as well. So how we verify this program? We actually compare manual tracking with program tracking. And this is the result of the comparison. So here's the length of each fibers which were detected and volume. And you can see that they're close to each other, and there is no significant difference when we look at average. So this program can be used across different types of cells and experiments. We tested it on mesenchymal stem cells and myoblast cells. Here's one example of experiment when we used the program in our lab. We harvested cells from mouse bone marrow and split them into group control and vibrated. So treatment was vibration in this case. After the vibration, we stained cell and highlighted actin fiber green and nuclear blue. Then we used confocal microscopy to take 3D image. Here we go, and then apply this program to reconstruct fibers and nucleus. Now, this experiment measured roughly 100 low-intensity vibrated cells, and here typical images from the control group and from vibrated group. They look different, but right now we actually can quantify it. And here we see measurements for these particular cells, and the algorithm indeed detected the increase in number of actin filaments on vibrated cell and increased number of branching points. This slide demonstrated the cell have a high number of fibers here and more branching points. And actually right now we prepare this data for publication for this particular experiment. There is just one example. This program can be used in many different types of research. In the paper with the method was published in Small Structure Journal earlier this year, and also the program is available as open source on GitHub. And I'm ready for questions.
Thank you, Nina, for this amazing talk. And we have time for one question. Есть время для одного вопроса, коллеги.
We have time just for one question, thank you. There are no questions, then I do have one.
The proteins like lamin A, like visualize structures of other proteins.
This is very good question, and actually this is where I'm living right now in my research. So the next step is adding exactly specifically lamin E and also we right now our nuclei has homogeneous structure, but we want to make it just to see how histones also located within nuclei, so it will be heterogeneous.
Mm-hmm. Thank you. Good luck with your research.
Thank you so much.
Mm-hmm.
Следующий наш доклад.
The next presenter, Gulnur Junusova, Institute of Genetics and Physiology, Almaty, Kazakhstan, please. The floor is yours, madam. Yes, there's a small announcement. I forgot. I will have the timer set for 14 minutes, and when you hear it, then this means that you have 1 minute sharp before you finish your speech. Thank you. Good morning, dear colleagues. Allow me, please, to present you the study. So it is TM receptors, early development of breast cancer in Kazakh population, the targeted DNA typing and sequencing. So this was done within the framework of the program that was initiated by the Ministry of Healthcare. We've been partnering with the Institute of Radiology and Oncology, and our project run by the Institute of Radiology and Oncology is conducted together with the Ministry of Science, and the prevalence and death expectancy from breast cancer is very high, both in Kazakhstan and worldwide, and therefore breast cancer is one of the leading types of cancers, and at the same time females starting getting this disease in young reproductive age, and the existing programs do not cover women younger 40, although worldwide the genetic reasons for inherited breast cancer as well as having cancer at the young age is something that has been actively studied, and in Kazakhstan we have just started those studies, and we decided to have a comprehensive analysis in terms of impact from genetic pathogenic markers in terms of development of breast cancer in young Kazakh females. And the studies included 224 women who had no relationships amongst each other with the primary diagnosis in the age younger than 40. We had AGS sequencing. We used MySeq platform to do sequencing. We also used the panel of 94 genes that are associated with cancerous pathology of different genesis, and it had 287 genetic markers associated with the breast cancer. And through-site sequencing panel can sequence the entire coding zone and the supporting entrons. So this slide actually depicts information in regards to the clinical and phenotypic studies in our cohort of patients younger 40, and this slide actually demonstrates that the youngest female was 19, the average age is 34 years old, and from the morphology standpoint, it was invasive flow type carcinoma, and the second is moderate differentiated and low moderate carcinoma, and histology has discovered aggressive forms and luminal H2 positive subtype. We have discovered, as a result of AGA sequencing and analysis, 23,000 genetic markers. So in order to filter the rarest genetic markers, we excluded population frequency markers with more than 1%. And what we have discovered was 59 pathogenic mutations, 38 unique ones that are frequently met, and overall it was 59 mutations in 57 patients, which is 25% of selection. And BRCA1 and BRCA2 would prevail, and in terms of the mutations, there were deviations in sequencing in reading. And the next is most frequently met pathogens and mutations that have been found in those patients. The table is very extensive, and we have discovered 7 new pathogenic mutations that were not found in other populations, and we have also validated our pathogenic mutations with the conventional methods of sequencing. And in terms of functional analysis, you could see that BRCA1 and BRCA2 frequently mutated. And in this very case, we would deal with BRCA1 in cases of cytosplicing, and so 52 and 66 was something that we would need in Polish, Russian, Ashkenazi, Jewish ethnic groups. So this is acknowledged as the founder's mutation, and that was something that we also have been frequently dealing with. And we also discovered the mutations that lead to shortening of the protein that creates 2 aberrant transcripts. That exclude the 21st exome and 8 nucleotides in the end of the 21st exome, which also get eliminated. And actually all of those 5 pathogenic mutations lead to the defects in terms of BRCA1 and BRCA2, also destroy the domains, and they disconnect with the other types of proteins as well, and you can see it on the slide. And also in our studies we wanted to spend less, because we are aware about the fact that what mutations prevail in young women, so we wanted to make a real-time PCR, and now we're preparing a test system as well as the panel based on the results that we have obtained as a result of those studies. So the design of primers was completed, and this slide depicts information in regards to the database verification, and you could see the mutations that were not registered so far as pathogenic mutations. And here comes the assessment of rare mutations and its impact upon the structure and functionality of the protein using in silico and software. So we have discovered 53 variants in 46 patients, to be precise, who were acknowledged cancerous with the 2 tools of focusing. And in terms of associations, in order to identify the impact upon the genetic pathogenic mutations, we identified whether they were the host body or non-host body specific. And in most of the cases, when it comes to host, the host of the pathogenic mutations associated with triple negative types of cancer, so twice as much it is risky for those women to have positive effect of treatment. And in case of the in terms of the focusing, we have discovered the consequences, and hosts would die twice as more frequently, and they would have the MTS or recidivism. And BRCA1 is associated with family inherited disease, and you could see the association OR3. I also would like to mention about the fact that we also received the results in terms of rare mutations, and over here you could see Manhattan top demonstrating the association. We have discovered two locus that were interlinked with each other, the polymorphins. So the first gene has 4 polymorphisms that are
polymers been attached to each other, and they are associated with each other. The force of attachment of first and the second one is depicted in the following results. The darker color you see, the more attachment we have. It's like 100% attachment. And on the second picture, from 80 to 100% of attachment. All the locuses which have polymorphism, they are associated with the breast cancer. We've seen it 3 times of high association level. We developed the methodological recommendations on implementing these results into the screening process, the breast screening national program. We would like to enrich it with the use of genetic analysis. Here you can see 4 options of mutations, different panels, starting from anti-shock till the multiplex, which covers 94 genes, general PCR, PR real-time, founder mutations, estimated founder mutations, which have been announced side splicer, for instance. 9409 only met in cases with Kazakh women, and in one Chinese population, 0.2%. So here we've had 1.8% the mutation of side splicing, 3% of occurrence among the young women who have breast cancer. Here we highlighted the high-risk group, the patients and their relatives with the heredity oncological anamnesis, the close relative family member with identified gene mutation of triple negative molecular subtype of cancerous tumor. And here you can find the key findings. We identified sequential mutations among the 57 patients. That is 60.9% from the whole group. 12 out of 38 different mutations occurred several times, and for those who had pathogenic mutation, they've had twice more risk of triple negative breast cancer developments. And thank you for your attention. Here you can find our key findings and 2 articles published. Thank you.
Please, if you have any questions, feel free to ask. Well, I do have a question. Yes, please. Thank you very much for a very interesting report. I have a question. Have you tried to look at the specific mutations in certain types of breast cancer? For instance, like inflammatory breast cancer. Did you have such samples? In terms of tumors, you mean? Yes, there's the breast cancer types as inflammatory breast cancer. Yes, the triple negative. So we would look at the blood. We didn't look inside the tumors. Do you mean the inflammatory? Yes, inflammatory cancer. Did you have samples? No, we didn't. Not the single ones. So, did you deal with these? No, we haven't. Nope. Thank you so much. So I do have a question, which is more general. So firstly, your paper is really important and relevant, not because it has to do with ethnic and cultural specificity, and it's so this is something that we need to look at. We have to know the specifics of regional diseases. So my question is, you mentioned about the increase in number of patients. So could you please comment on that? How many fold? You probably have the statistics in Kazakhstan, or starting from 2004, breast cancer remains position number 1. So what was the growth in fold? I don't remember exactly. Yes, but it is getting increased. So, for instance, autism, right? There's such a disease, and lately, for the last 3 decades, it grew 5 to 7-fold, and it cannot be related to genetic mutations only. And the second point, the mutations that you have discovered in the associations, are they somehow associated, with something else, apart from genetics? Could you please make a stress on whether it is belonging to Kazakh ethnicity? Well, actually, the different tribes, ords, and so on, that's a different question. So specifics. Yeah, yes, indeed. What did you discover? Thank you so much for your question. This is a very relevant question, and we try to apply for a new project, which is something that we hope to get results from on specificity of those pathogenic mutations. The frequently found pathogenic mutations is something that is associated with the haplotype analysis. We can prove actually that there is a founder's effect, in given population or in geographic region, for instance. Those mutations prevail, right? So this is called the founder's effect. So based on STR locus, we proved that those were the founder's mutation. However, I didn't make those announcements because those articles were not published. Actually, they are specific, and associated with the breast cancer. Why? As they breach the function of the protein. Thank you. What are the reasons? What factors could lead to breast cancer? ethnic component, as I said, and besides, those are the factors. So hormone independent environment, smoking. So late pregnancies and labor and so on. Thank you. Thank you so much. If you have questions, you may approach me during coffee. Thank you. Allow me please to give the floor to the next presenter from Nazarbayev University, Marina Kryzhevskaya. Please, the floor is yours.
Добрый день, дорогие коллеги.
Good morning, dear colleagues. I would like to appreciate the organizers of this conference so that I could share the data. I would like to apologize that the slides were presented in English, so today is my experimental report. It is going to be delivered in Russian, despite the fact that the slide is in English. So actually, we look at the bladder cancer and pathology of bladder cancer. We've been looking at the mechanisms that could be targeted while treating the patient. And you know that at the moment, it is a general position that there's a epithelial mesenchymal transition, which is a driver of tumor progression, and it has to do with sustainability and reactions to treatments, to include chemo and resistance to chemo, and the immune response is also controlled by those mechanisms, and they may occur in single cells, and you could see the primary tumors. So we get to deal with single cells with a specific development of and progressing of cancerous cells. And then later on, at a later stage, being exposed to different treatments and specific therapy, those cells are not prone to pressures from specific drugs, and then there's drug-tolerant persistence, right? And as a result, those tumors are resistant, and there's a secondary metastasis process. And bladder cancer, in fact, is pretty unique. Why? As this is something that gets registered at the later stages, and that cancer in particular could be separated into two parts, so which is curable and non-curable. That the lethal effect, we have data starting from 1992. The death levels remain the same, and the level of disease is getting lower. Why? Because patients now are concerned about the environment. Predominantly patients actually watch the diet and so on, and this leads to the decrease in patients. In terms of the life expectancy in bladder cancer, you could see that in case of his MTS form, then the life expectancy is around 10% of patients. But in Kazakhstan, it is much lower, about 2% in total. So the question is, how could we actually have better performance, and what can we do with that? This slide demonstrates different types and stages of bladder cancer. You could see that there's so-called carcinoma sita, which is treated pretty well. It's actually enough just to have a resection, and then those types of cancers could be actually controlled well. However, for the patients it's a complex procedure. Why? Because they have to go through the internal checkups on an annual basis. However, when we deal with the more advanced cancers, it's almost not possible to help a patient, and the life expectancy worldwide is no more than 5 years. How can we treat bladder cancer? Depending on the stage, the global practice is based on chemo resection, radical resection of bladder, and when there's more advanced stage, then the total reconstruction of bladder takes place. Those stages don't exist, and it's just platinum protocol is used, and immune therapy has been commenced back in 2020. So the question is why certain cancers could progress to later stages, although initially they look as pretty good types of cancers from the prognosis standpoint. You know, if we know the targeted genes that will help us actually to block the activity of cancer with the help of indications, we could provide help to the patients. And this is really relevant for patients from such countries as Kazakhstan, because the reconstruction of bladder doesn't exist, and 6-platinum is used only at the earlier stages of treatment. We have been closely looking at the tyrosine kinase receptors that get specifically activated into cancers. Actually, those receptors are the receptors of phytocentric system. They actively get engaged in macrophages, and they eliminate the deaf cells from the tissue cultures. And our interest, since we are dealing with epithelial transition, we focused on AXL gene, and you could see the results of the simple expression of this gene in different cell lines. And in case of bladder cancer, that gene gets activated, and mesenchymal subtype of bladder cancer, and they have the characteristics of the stem cells. So should we make the analysis of Kaplan-Meier, the combination of high and low expression and TYRO3 is associated with poor survival of bladder cancer patients, and AXL receptor gets correlated with poor survival of patients. Now allow me please to demonstrate the structure of those receptors. So you could see the 3 domains. So extracellular domain, that could be proteolytic, cut, and it could be found in urine of the patients, and transmembrane domain, and internal cytoplasmatic domain. I'm now talking about receptor tyrosine kinases, and you could see the signals that is delivered to other genes, and the mechanisms get activated, because normally they don't get activated under the normal circumstances. So these things associated with the cancerous progression. So we started this work as follows. The detailed studies of the receptors was something that we began working on in Kazakhstan, and we started with the purchasing of slides with multiple samples, and over here you could see 75 samples that were repeated, and those are the samples who were diagnosed with the bladder cancer, and there are different forms of bladder cancer, and when you purchase such slides from Biomax company, you could actually assess the condition and the status of receptors. In case if you are interested in factors that could lead to bladder cancer, then you could have a closer look at the specific types of bladder cancers, and as the full-fledged characteristics for patients is provided. So what we have discovered is that the specific expression, and of course those receptors could be expressed altogether, however there is reversed expression as well, and we decided actually to have a closer look at how the control group could perform. For instance, we could actually compare the expression. Over here I would like to give you the inverse correlation examples between AXL and other receptors. This is very aggressive form of bladder cancer, and you could see that it is pretty specific in terms of regulation. So this is something that tells us if we're going to be having specific targeted therapy, knowing the sensitivity of the tumor of the given patient to the given medication in the given circumstances. In this case, we're talking about the epithelial transition. Then we could actually impact upon the destiny of these patients. And how can we do that? Of course, you could do that with the help of the same immune histochemical analysis, and sensitivity is really hard to identify. Over here, I would like to share information in terms of expression and the correlation of TAMs in bladder cancer tumors with the specific, very aggressive forms of bladder cancers, like mucous adenocarcinoma, carcinoma, squamous adenocarcinoma, and so on. So the cancers that are almost non-curable. And in 2023, Almaty team of experts has published info in regards to 5-year life expectancy starting from 2017, and they demonstrated that the life expectancy is really, really low. For instance, when the patient is diagnosed with bladder cancer, for instance, adenocarcinoma of the bladder cancer, then that patient may not survive. That's absolutely not possible, subject to current therapies. And the question is how to study these. Yes, in parallel, we have looked at the activation of those receptors, and the correlation of the status of those receptors is ongoing. For us, this means we can provide the treatment for the patients with the help of the studies that we've been conducting so far. We also study the protolytically healed fragments, and this was something that was found in the urine, and this is a opportunity for us to diagnose and study bladder cancer. And in case of specific expression of TYRO3, we can see and for us that's a opportunity to have a closer look at the tumor of the patient. Instead of having comprehensive analysis, we could actually have the liquid biopsy to have a look at the urine of the patient. And in parallel, all analysis or all lab work gets verified with the protein analysis, Western blotting, and we testify that it's not the error, and protein indeed is found in the urine. My time, so 1 minute, okay. And this is the most interesting part. So actually, I'm now demonstrating the conventional method of having 3D cultures, and they are totally reflecting the genetic status of the tumor. And you could see 3D cultures that remain unchanged. And by doing so, you could actually test the samples of patients. And starting from 2021, we grew those samples. Could you give me 2 more minutes? Yes, we can. Thank you so much. So actually, starting from 2021, we started growing those samples, and now we can treat them with different substances to include cisplatin and specific targeted substances, and we see the analysis of expression of those proteins. Unfortunately, not like other labs, we are still at the level of the fifth passage. We can grow the culture, but we cannot make the library of those cultures. And we want to use receptors and using the medications that are well distributed, such as verteporfin and metformin, actually may have an impact upon the expression of the receptors, and we want to have a look at how they impact upon 2D in Petri's pan, and our intention is to have a look at what is going with those receptors in primary cultures as organoids that I have demonstrated to you. So the last achievements in science and 3D culture is development of urinoids. This is something that we've been very actively engaged in. We managed actually to have stem cells from urine samples of the patients. We grow them, and by doing so, we are capable of making diagnosis. And before the patient has a resection, we could actually check the sensitivity and characteristics of the given tumor. I would like to thank all of the participants to this project, and give special thanks to Mr. Ogai, Director of National Center for Biotechnology, because he's been assisting us hugely. And I also would like to thank our colleagues from the Institute of Oncology. Thank you. I will ask you a question, and then you will answer during the break. Okay, so this is going to be my punishment. Thank you. So actually, the tyrosine kinase as a biomarker was mentioned, and the second, so how many years ago, was it proposed? So this is the first question, and the second question that you will answer during the break. 10 years ago, I participated to the international project on biotargeted medication, and that was an EU-led project. So the conclusion was that there are many biotargets, there are no medications. No need to respond. No. I will. No. Thank you. You will respond during the break. All right. Then we will continue later on. So remember, I started with AMT. We need to have the signature progression. Thank you. Novikov Dmitry Georgievich, Omsk State University. The floor is yours, please. Good afternoon, dear participants to the conference. I would like to congratulate National Center for Biotechnology and on behalf of our university to wish you success in your endeavors. Yesterday, they mentioned about Omsk as the homeland of rare strains of tuberculosis, and this is something that we also are hugely engaged in, but let me talk about pathogenesis. Since I work for the medical university, I would like to say that traditionally the students of the medical university start studying TB based on the concept that this is the the role of neutrophil white cells while looking at the pathology is almost not studied at all, and that has been the situation for many years in a row, right? And one of the most recent reviews demonstrates the off-cell traps. And why is it actually left without any attention? Because we have to mention about having off-cell traps of neutrophils, and it was opened 20 years ago, sharp, and the basic functions on generation of active forms of oxygen, and actually the nithos is the process that was accidentally found by the Max Planck lab, and off-cell DNA gets dispersed to the extracellular space. This is a physiology-based process, and trapping means being able to catch the pathogens. So, what does the trap contain? Whatever this is, DNA of neutrophil, white cell, the histones that would fix it, and the components, the granula of protease, bioactive substances, and whatever has, an opportunity to eliminate the pathogens. So what are the stimulators, ranging from microorganisms all the way down to the molecules of elimination, and different activators, protein kinase, for instance, ionophores, and even the crystals, cholesterols, and urates. And of course, experimentally, whether neutrophil is able to survive, yes, it may. There are two forms, vital nethosis and suicidal nethosis. It's not necessarily for this substance to die. No, it could be. We don't need protein kinase, but we need the ions of calcium. In order to have the components of the nuclear destroyed. So in fact, actually, the differences in between vital and suicidal types of nethosis is that this is the classics, right? in practice, the way it gets interpreted is that it's a very complex task, actually. Why? Because using non-typical stimulators, over here we talk about the two types of stimulus, using ex viva conditions to stimulate, it's very hard to differentiate between those forms, actually. And one more aspect, should you need to have a pool of off-cell DNA. I'm also referring to the reports given earlier, and the histone H3 is losing its positive charge, and then thanks to the arginines in this process we get the triple enzyme complex participating into the process. So this is the enzyme that was getting the most attention. And when it comes to mitosis, the researchers can't come to a consensus on different aspects. The authors have been looking at options how to interpret different phenomena and the different mechanisms of mitosis, to include mitochondrial ones and so on. So how to study? It's really complex. There's no golden standard. It never existed for 20 years in a row. You could use electronic microscopes, flow cytometry we mentioned about yesterday, and IFA methods, allowing us to reveal some of the elements. So we use the classical approach, which is based on fluorescent microscopy. And here comes our lab, and the methodology that we've been using is having the classical approach demonstrated on a slide with non-specific stimulation, and further visualization of the objects is done thanks to intercalated dyeing agent and XCD antibodies that the neutrophils have. They are marked with fits, and actually this method could be patented, and we patented it. Earlier the dual branches were not used. So the logic is that the standard dyeing agent for flow cytometry is used to assess viability. It gets linked with DNA specifically, and in our case linkage with DNA is the target, right? And so appearance of DNA in extracellular space, why can we say that's for sure? It goes beyond 15 feet membrane. So this is something that we want to see. So there's no mitosis, so neutrophil is intact. So here comes the activation, and we have identified different forms of activation, and you could see the condensation of neutrophils, and the membrane is still at its original place, so going beyond the locations, and you could see the fragments of the membrane and a couple of neutrophil traps, so actually here comes the non-specific stimulator. So you could see the location, and the size is large. So and here comes the actually overall summary of the neutrophils, and so we are targeted at TB. And in children population, it's not very actually widely spread process. However, we are concentrated on the immune response in children. So the question is, why have we been studying the neutrophils? The neutrophils actually in the in the off the track space is something that we're not going to be able to cure, but our able is to make sure that the TB is the systemic disease, and cytokines pro-inflammatory, the certain phenotype, and of course we have to take into account the overall mechanism, and this is the process that we've been studying. So those are the groups of children. Of course, we've been studying healthy children and those children who have lungs pathology and those who have TB. And while comparing control group with those children who have latent group of TB, they have just positive reactions to TB antigen recombinated, and they have both thread-like and cloud-like groups of LTI ATP+. So potentially the neutrophils in blood of these patients has been altered. In regards to other forms of activation, there's almost no difference, and the number is growing in case of TB, you know. Why? Because the entire set of neutrophils was used for nethosis. So whatever refers to tuberculosis of respiratory system, you know, there were differences in terms of clouds. However, the thread-like traps were not different from the TB in respiratory system. However, the respiratory TB was heterogeneous apart from the other forms of TB. And the further separation and studies demonstrated that, in fact, should we separate it into two clinical groups, the classical primary TB complex and TB of lung lymphatic nodes located in sternum, we could find, neutrophil traps in both of the cases. And the thread traps in case of TB of lymphatic nodes are not generated that successfully as in the first group. So, moreover, in order to verify our concept, we had had studies thanks to IFA, the structured citral cystone H3. So what we have found was that this is something that we frequently meet compared to the TB of lymph nodes located in sternum. So netosis have already happened, and this is something that has been circulated in the blood. And there was a correlation, a positive correlation between thread netosis and the level of cystone H3. So we have to mention about the molecular profile of TB compared to the primary classic TB and the TB of lymph nodes located in sternum. So actually, in fact, that's a association to the pathology. In fact, there are almost no publications on cloud-like traps, and there was only one publication that we managed to find in immunology magazine, and so it was very short note in regards to making a notice of different types of traps, and actually they call it that those traps, they have to be demarginalized, and nethosis is something that has to be, and this has to be called the forming of immune traps. And again, so in fact, at the molecular portrait, this is how the TB in lymph nodes located in sternum looks like. So we could actually then make the conclusion in regards to antibodies formation. Thank you so much. We have time for one question. I will ask it. So short question. So do you somehow refer exosomes and vesicles? Do they somehow, correlate? So actually the question is how do we form the vesicles, right? So there are lots of sources for extracellular vesicle. Okay, so actually in fact vital necrosis could be related, to the formation of vesicles. There's no, rupture. There's just packaging of membrane into, given clusters. So again, going back to hemostasis, and the vesicles is the object of interest. And from this standpoint, actually, we could. I won't be able to demonstrate you that, and from that position, we could actually mention that necrosis and hemostasis are pretty interrelated. We talk about the clotting, and there is a correlation in between micro vesicles, and it could be associated with hemostasis process as well. So any more questions? So the question, you may ask the speaker your questions during the coffee break. And now the next presenter, Alina Korovkina, she's going to be delivering about the digital PCR. Please, the floor is yours.
Добрый день, коллеги.
Good afternoon, dear colleagues. I would like to thank the organizers of the conference for such an interesting program. And I would like to present in regards to the digital PCR. And to start with, I would like to actually tell you about the method itself. And the third generation PCR is an absolute quantity-based method, allowing us to have the absolute figures of RNA and DNA in molecules. Actually, the digital PCR uses the endpoint, and the real-time PCR has inherited, just the name. In fact, digital PCR uses the same fluorescent zones or intercalated dyeing agents in order to identify the molecules of nuclein acids. And PCR is digital PCR is compared to quantity-based PCR. And in this very case, the digital PCR is an absolute method of quantifying the samples, because the sample gets distributed evenly on the wells, and there could be thousands of those, and in fact the sample in parallel is run in thousands of wells for one sample. So the idea is that the targeted molecule either is going to be getting inside at nano wells, and where the targeted molecule would be synthesized, and This fluorescent signal is also going to be synthesized, and you will have a look at the slide. And there's going to be the stars sky demonstrated on the screen. So each of one of them is the molecules. So the digital PCR doesn't need to have sampling with the known concentration, and it is more and more precise compared to the quantity PCR. So in order for digital PCR to be possible, a company called Kaizen has developed the nanoplates, and you could see the nano reactions and nano wells demonstrated, and I have demonstrated the scheme of the plate, and each and every sample has the complex well-based structure. So depending on the plate, the nanoplates could be either 8,500 or 26,000, and that's purposed for more sensitive studies. So it's better to see once than to hear 1,000 times. So I decided to demonstrate to you how the system looks like and works like. So the optic fiber is covered with the rubber, and inside the machine you could see that the plate goes inside the module with 96 columns pressing over the rubber, and the primers in the sample gets pressed through the well-based plate, and then here comes the nano reactions pinched and blocked. They become independent from each other, and then the plate gets transferred over to the modification block. So the digital PCR is the detection based on the endpoint analysis. So that's a simple amplification. And actually, in this very case, the sample is distributed in thousands of nanoreactions, where we would have the targeted molecule of DNA or RNA. The signal gets amplified, fluorescent activity gets intense, and we distributed it in thousands of nanoreactions, and only where the target has managed to go to, the signal is going to be amplified. And then in very same machine, it goes to the detection block, and we identify the signal dependent on the intensity of the backup response. So we do have different types of PCR systems, and there's a whole spectrum of machines called Kaikunity. So they're classic real-time amplificators, and they could have different channels of detection, either 2 or 5. However, there are machines for wide spectrum labs where you could have 4 to 8 plates analyzed at the same time, and plus the digital PCR is not just more precise, but together with the sample, there's an opportunity to have a random distribution of molecule inhibited. And digital PCR is more sustainable in terms of in vitro. We're going to be mentioning about the application of digital PCR, and in this case the spectrum of methods and the spectrum of applications is much more diverse. And I wanted to talk upon biomedical studies, and I would like to mention about having digital PCR in therapy based on analysis of adenoviruses. You know, it's one of the most popular types of treatments and one of the most successful treatments. Perhaps you heard about Zolgensma, and this is based on virus therapy, and it is purposed for spinal muscular atrophy in newborns, and this medication is known as one of the most important, expensive medications in the market. You know, one dosage is costs up to $2 million, and actually the adenovirus vectors and compared to CAR-T, for instance, adenocarditis virus do not change the DNA of the patient. All they do is they deliver DNA and infect the cell. And actually the DNA is the same. And the protein is going to be generated without any alterations. And of course, the positive characteristics is that there's a wide spectrum of adenoviruses. However, there are some limitations and restrictions. And the restriction is demonstrated to your left. You could see the size of the genetic structure. It is limited. It is just 4.7 kilobase. And in case if you need to transform a large structure, it's not possible to do. Now the studies are conducted allowing us to enlarge the size. And it's just the ITR elements that are left after the virus. It's CAR-T repetitions, and the internal part actually gets the coded sequence. And in this very case, while forming such genetic constructions, the virus actually absorbs DNA, and while packing the virus, it could not actually pack the entire genetic construction. And the digital PCR may be used not only for the quantity characteristics, because it's really important to calculate how many vector units there's going to be in one dosage of medication, because different patients have different size and age and so on, and characteristics. So therefore, now we have to verify whether it is going to be packaging process arranged correctly or not. It is done elegantly. So I'm talking about the targeted sequence, which is ensured in a couple of zones, and there is a chance to look at the digital PCR in 2D to verify whether the necessary targeted paths inside one nanowell. And over here you could see the classical image of digital PCR. Please pay attention to the oxiles. There are two clouds. The blue one is positive nano well with amplification and fluorescent signal, and the gray cloud with this is the negative reaction with no DNA that penetrated the well. And you could see two graphs, and the dark blue are the nanowells that have two targets, promoter. And should we actually place all of those pictures on the top of each other, we could actually identify the number of associated carcins that actually demonstrate the entire picture in terms of vectors. So the next is qualification during biologics manufacturing, and so in this very case, the question is, so should we have cells that we have to use for working with CAR-T, for instance, and lentiviruses. After the product of protein is complete, we have to clean it up, and in the end, for safety, we have to verify whether there's a leftover DNA in the medication, in the product or not. So, and the digital PCR uses its privilege meaning that it is more resilient to inhibitors, and overall the product is entire set of interferal system, and this actually allows us to see the leftover DNA of the host. And we prepared the kits. The first one is Chinese hamster aviary cells and human being cells, and we actually demonstrated that the sensitivity stats were 5 femtogram. You know, it's a very, very sensitive system, and in fact it is very inhibited. So to complete, I would like to tell you about one more application of this biomedical product. And actually, mycoplasma are common contaminants of cell culture, and mycoplasma actually has a very small size, and while making a biomedical device, it's really important to check for mycoplasma contaminations, because it can lead to infections in GU and respiratory tract. So therefore we have to make sure that the equipment and machines are clean. You know, we have to check the levels of mycoplasma before we make the final product. And in this case, company Kaizen has developed a special kit for digital PCR that also could be used very actually successfully, and this is a mix of proteins that may inhibit the reaction. So you could see that despite the fact that the internal control in the inhibited system could be lower than the expected one, and the tolerance to sample matrices is ensured by the fact that the system is interferated. Mycoplasma detection has a high precision and accuracy, as the toolkit has the primary sensitivity at very high levels. So that was my last slide. So I made it through. So thank you for your attention. And if you have any questions in regards to digital PCR, or you need to make the design of your experiment, you could find me. Please join the stand of Vienna. So this is our partner in Kazakhstan. Thank you so much. So before the coffee break, we have one more presentation. So Mr. Dubaeva, Elena Vitalyevna, please the floor is yours. We'll be grateful if you could take up to 10 minutes. You know, I will try to even do it faster, so we could continue during the coffee if you wish to. So the topic is genetic factors of risk in brain tumors. Actually, you could see that 10 reasons for death globally, and item number 3, which is strokes. And looking at the global map in terms of death rates in 2021, this was something that was added as with the data as of 2024. So again, please let's have a look at Kazakhstan. Unfortunately, Kazakhstan is characterized with high levels of death. So therefore, you know, what I would like to say is that the studies in stroke and the nature of stroke is relevant for Kazakhstan. Now let's have a closer look at stroke. There could be ischemic and hemorrhagic. So we are more interested in hemorrhagic stroke. Why? Because it leads to 50% of deaths, and those who suffer from that disease in most cases range from 30 to 45, and as a result of the hemorrhagic stroke, the likelihood of having disability is very high, and one of the reasons for hemorrhagic stroke is that there's arterial venous malformation in brain. So what is it? Which is a bundle of arteries and veins. So actually the vascularity is not normal in patients, and this leads to bleeding. So I have to mention that before AVM actually was diagnosed, and before having MRIs, they thought that this was something inherited, And actually, before they started to get the analysis, then they came to a conclusion that those were the sporadic forms of AVM. So this is caused by polymorphism that may lead to the risk of development of AVM in brain. And back in 2018, there was a hypothesis made by Nikolaev and colleagues who have said that somatic mutations may lead to development of AVMs in brain. So consequently, the literature review that we made was something that we wanted to verify in our population in regards to the polymorphism, identifying the risk of development of that disease. As everybody knows, that ethnicity-related markers are relevant. So we work together with our colleagues from the neurosurgery university who recruit patients for us and are responsible for the entire set of diagnostics. We could see the characteristics of individual patients who had AVM in brain, and we have taken Kazakh population, so ethnic Kazakhs only, and we also had a control group chosen by our colleagues and with no malformations or aneurysma. And one of the criteria was that AVM of brain and the scale of marking is one of the clinical indexes used by neurosurgeons while prescribing the surgical treatment for patients. And next, should we have the S&P sampling? Now let me talk about the sampling that we would collect. There were different approaches. We would deal with tissue and blood, and our objective was to have a closer look at somatic mutations. So just 8 samples in tissues, and why we did so is that it is the tissue or brain that is very hard to get collected. Only those who are recommended to have embolism of vessels, they do have sampling prescribed. And the last column is this crash status. We wanted to check the hypothesis that was mentioned before. Activated somatic crash could actually participate in the final mile. So There's no statistics that would be published back in 2018. Was not available. So in order to have SNP genotyping and studies of the SNP markers, we decided to make the targeted sequencing, and we picked not only the considerable polymorphisms mentioned in literature reviews, we took the genes having an objective actually to since not we could potentially find something so that we could make a presentation of the targeted therapy and targeted panel.
With the help of ingest sequencing, and if you will take a closer look to the listed genes, you can find out that some of them are included into the pathogenesis, connects with the blood vessels, arterioles, and veins. And it also includes inflammatory factors, fibroblast growth factors, as well as and other factors too. And I will not show you the complex table with the whole list of polymorphs, because not all of them have a statistical value. In most cases, statistically valued being photolicking and MMP9. In terms of statistical value, we could also add APOE. And in our publication, we mentioned that this particular part active in the process of polyprotein attachment. We used already done testing on cross mutations. We have other approaches too in terms of identification of somatic mutation and the normal mutation, including sequencing of exome in blood. It sequences blood and tissue, and as a result, we get an exome analyzing samples. Here you can take a look at an MRI of the patient. An arrow points to the malformation EVM. As a result of exome sequencing of DNA, which we get from blood and tissue, we receive the VCA file and its annotation filtration. We got more than 30 genes, following up with representative analysis using enrichment analysis and OR analysis, and we found out that the gene, cysteine gene, is statistically valued, which takes a part in the foxome. And it is statistically valued for the article, and there is a need to prove that the polymorphism which we got undergone all the analysis and verification. We verified it. Here you can find the comprehensive map of the signal way. And currently we have more and more publications within the last 5 years on analysis of pathogenesis in relation to AVM using the transcriptional analysis, which identifies the transcriptors, and we use endothelium tissue in a normal condition and the tissue with EVM. Here you can find not the complete results we got. We continue our analysis of the following samples. Here you can find only 3 of them, and it's seen that the differentially expressive genes are high in terms of number. We use the increase of expression, decrease in expression. We have different bases, and one of them is TICOPAF, which identifies which specific genes take a part in this process. You can use the Nikolaev article and find out that the most somatic mutations leads to the AVM are associated with the signal pulse. And please take a look that the most parts of the differential expressive genes are active in signal paths. That's it. Thank you. Once again, I would like to repeat that Elena Vitalovna will be available during the coffee break. You can ask any questions you want to address. Now we'll have our coffee break, and we will continue exactly at 11:30 because we have the REACH program. And I would like to address one question in advance to the speaker. Have I understood right that the stress realized through the different signal systems send it to the brain in certain situations could lead to the deterioration of functioning of the signal systems and the diseases you mentioned, because 97% that is the paradigm of polymorphic genes.
Коллеги, через 2 минуты начинаем сессию, то есть продолжаем. Коллеги, проходим в зал. Кого? Да. Вот это вниз.
Ну, коллеги, давайте приступим к второй части.
Dear colleagues, let us commence the second part of today's meeting. And in fact, the first part is something that we have started with, so during an hour, we now have 60 people in the room, so we are more successful. I don't know how many people we're going to be getting by the end. Should I be in Russia, I would, offer the Ministry of Science to recommend that in the beginning of each section, we would have the representatives for each important section, so we would not need digital PCI amplification. So actually we're going to be having the report to be made by Pavel Viktorovich Arlikov. You could see the title on the screen. So for me, those topics are very, very interesting. So I participated in those developments for the most recent 15 years. So I'm interested in listening to your report. So thank you so much. So dear colleagues, allow me please to provide you with the preliminary results on the current project. We're going to look at the status of genetic diversity of Mycobacterium tuberculosis in Kazakhstan with the help of polygenome sequencing. I just wanted to remind you about the relevance of this disease, and actually many people think that TB is not about that individual, and there's no chance to actually get exposed to TB. However, statistics tells us about the contrary, and the most frequently killing infection is TB. So a couple of billion people are infected with TB, and actually infected doesn't mean ill. And this picture could be presented in the form of an iceberg. When infected get sick, And TB actually is the reason for 1 million of deaths every single year. And we could have a comparison in 2021. COVID actually has hit this record for a short period of time. Now TB is #1 reason of death, and we have lots of cases in children as well. And about 10 million people get sick every year. So where does tuberculosis come from? And in accordance with the retrospective data, it is known that it existed in ancient Persia, in ancient Egypt, and ancient Greece, and in 7 out of 28 mummies that were buried 3,000 years ago, had the traces of TB. So around 1/4 of the most richest people actually could have the respiratory form of TB. And Hippocrates and other prominent ancient scientists would write about this. So where does it come from? We are aware about article by Inaki Komas, and around 70,000 years ago, the human being actually delivered TB from Africa, and TB got distributed throughout the world. And in fact, actually, such interesting facts such as mastering of fire played its role, actually, and there's an article published in PNAS saying about the fact that an ability to have fire was the factor that distributed to exposure of TB all around the globe, and that was a conjoint evolution of both human being and bacteria. Overall, speaking of the disease as it is of TB, the situation in Kazakhstan is more or less fine, and Kazakhstan is not marked in dark green color, illustrating high rates in diseases. We are light green. So in terms of other characteristics, then the situation is a bit different. And should we compare prevalence worldwide in terms of years, in Kazakhstan it is getting lower and lower, gradually, slowly but surely. And again, such a parameter as the number of patients who got sick with multiple resistance, then it is not that great. If worldwide the situation is getting better, in accordance with this criteria, in Kazakhstan, on the contrary, this indicator has been worsened, and this is a reason for us to concern. In accordance with WHO, Kazakhstan is one of the 30 countries with the high levels of antibiotics-resistant TB, and Kazakhstan is marked in dark red color, so the situation leaves much to be desired. And at the same time, TB incidence is a hard burden for the state, as treating the patients with such form of TB is really expensive, and previously it was estimated at the level of 2 million tenge. Now it is much more expensive per year. So let us think about why we had this situation in Kazakhstan and in neighboring countries. We have to take into account the genetics more thoroughly, the genetics of the pathogen. So what genotypes prevail in Kazakhstan and how they get evolutionized. We are interested in bacteria resistant to medications. And in fact, We had a large-scale study, and it was published in 2019. We studied 700 mycobacteria of TB, together with our French colleagues from lab represented by Elena Vitalyevna, and based on those results, we understood what genotype and what line of mycobacteria prevail in Kazakhstan. And in fact, it was something that we mentioned about previously, and the Skiba, Akhmetova mentioned about those facts as well. And eventually, 80% of isolates refer to Beijing family, and around 20% refer to line number 4. So it's LL4 3-lam. So whatever refers to the global distribution, the picture looks a bit differently globally. So line 4 prevails, on the contrary, not as in Kazakhstan. So should we actually look closer and have more detailed classification and subtitles? Back in 2021, classification made by Travarnatan was published, and I think this is something that is widely spread and accepted in terms of L2. So he has described subtypes and Beijing strains, and we were actually interested what strains prevail in Kazakhstan. So we built a distribution panel in terms of the neighboring countries. We were interested what subtypes would be dominant in terms of subtype L2, Beijing, and so on, and in neighboring countries. So you could see the turquoise color that is compliant to L11 genotype 4. So in order to understand in more details the structure of the population, we made sequencing with the help of full genome sequencing machines. So we took the same samples from the same regions, and we made full genome sequencing using aluminum MySeq platform, and you could see 126 isolated substances, and now it's much more. We didn't have time to analyze them completely, and we worked thoroughly in Astana, Ataral region, Kostanay region, Kizilorda region, and so on. And should we have a look at the subtypes, how they got distributed amongst 126 samples throughout Kazakhstan, the most frequently met is Central Asian outbreak. It's about 67% out of all of the samples. Number 2 is subtype Central Asia. Number 3 is W148 outbreak, which is well known to our Russian colleagues where it prevails, and other samples were presented in less numbers. So actually half of all samples actually would have multiple resistance to medications. So around 20% would be the substances with multiple resistance. And we tried to analyze whether there were some publications in regards to the neighboring countries, and our German colleagues made a publication on multi-resistant drugs in Central Asia, and they were describing the isolates in Uzbekistan, and it was no wonder that the population of India isolates in Uzbekistan would prevail, and Central Asia outbreak would prevail in their case as well. So in regards to W148, as I have already mentioned, in Russia, this is the prevail isolates, and they're prone to multi-drug resistance. Then we made the phylogenetic analysis, and we took one of the regions, Astana, and currently Astana is sequenced, and we took it as a representative one, so that we could demonstrate how the structure of the population looks like. It is similar to other regions, by the way, and majority of isolates were represented by Central Asian or Central Asian outbreak, and we have clade A W148 as well. And what evolution prevalences and properties do those isolates have, why they become so resistant? This is not invented by us, this is a well-known fact. Compensation mutations actually impact upon that. The Matthias Merker in his article mentioned about this tendency, and having the multi-drug resistance in terms of distribution of isolates is linked to compensatory mutations. This is something that is associated with the multi-drug resistance. So those mutations are based on the mechanisms that allow us to keep viability of bacteria after mutation is found in RNA polymerase, for instance, the most important genes. The mechanism of impact is demonstrated on this slide. Further, we decided to have a closer look at the compensatory mutations, and should we take Astana as a region, there were a lot of mutations of that nature, and around 90% of samples would have compensation-based mutations, and they were related to incoming mutation in POBH 400 gene. 408. And besides, we looked at compensation-based mutations that were studied in the article from our neighboring countries, from Uzbekistan to be precise. So there were basal line mutations, and they were identified as the founder mutations, mutations for strains found in Uzbekistan. So out of 126 samples, we didn't find almost mutations of those kinds. And going back to the analysis of those compensation-based mutations in terms of prediction, for instance, the so-called soft dynamode that is capable of making the prediction of free energy of Gibbs, entropy, and so on. And having estimations made, we came to a conclusion that the majority of them are positive, so it may potentially lead to stabilization of units of polymerase, except for one mutation, so exchange of amide acid in position number 448. So in terms of the search tool, we actually came to a conclusion that this is the sample of a convergent evolution. So therefore, it pops up independently in different isolates of mycobacteria. And from the energy standpoint, it is not that beneficial for the molecule. And at the functional level, it could have some evolution-based benefits. And we were interested in having the structure of population described in more details, and together with our French colleague, Christophe Sola. We used his platform called TB Annotator, allowing to calculate S&P metrics, and it can do much more, you know. It could predict polygotype, the resistance to drugs, and so on. And we received information in regards to more detailed clusters in terms of the structure of the population, and what we have discovered was that we found clusters that are different in a small number of SNPs, less than 12, which is evidence of direct transition. So we found some local outbreaks, and we also offered some additional genotypes in the families of the modern B gene. But again, those are the preliminary data. I'm not going to be commenting more on these. And currently we keep going, and the conclusions are demonstrated on the slide. I'm not going to be reading them. The only one thing that I would like to mention is that most likely there was a separation of two populations in regards to Central Asian outbreak, and Central Asia, Kazakhstan and Uzbekistan originated populations, and most likely it happened before the USSR collapsed. And in order to have this testimony to be approved, you know, we gotta make an additional analysis, which is due shortly. Thank you. One question. So Central Asia, Kazakhstan, Uzbekistan, so why and what factors led to the fact that you now have the new traits and differences? So what factors would lead to this situation? What is the difference between Uzbekistan, for instance? Southern part of Kazakhstan most likely has no difference from Uzbekistan whatsoever. So what are the factors, if any? Can you make the assumptions? Well, in fact, the picture more or less looks the same. You know, we have two clusters, they got separated, and in fact, actually, my anticipation is that there was an evolution-based benefit, and back then it was very complex time, you know, collapse of USSR. Perhaps the conditions in Uzbekistan were a bit more complicated than those we experienced in Kazakhstan, and perhaps over there they had more favorable conditions for TB. So the decrease of immunity, stress, you know, lack of food, you know, and insufficiency in medications, and so on. Thank you. So there could be many factors, actually, perhaps. Thank you so much. The mic is not being used. Interpretation is not possible. Thank you. So the next presentation is going to be the continuation of the same topic. However, it is going to be so application of target sequencing in routine diagnostics of TB. Nikolai Bubiev, executive partner, account specialist. Thank you so much for your introduction. So, dear colleagues, so firstly, I would like to congratulate you with your anniversary, and this is a very meaningful date, 30 years in a row, and so thank you so much for being so hospitable. And I would like to thank Pavel for fundamental work that they have conducted in terms of phylogenetic analysis of TB bacteria. And firstly, I would like to give a teaser in regards to conflict of interests. I represent a commercial organization. However, I will try to make education-based presentation, and we're going to be talking about using targeted sequencing in clinical practice. The huge amount of work that has been completed by the scientists, to include those working for the National Center for Biotechnology, led to the fact that we managed to have targeted panels aimed at clinical application in terms of diagnosis of multi-drug resistance in TB patients and having it used for targeted therapy. And that actually led to production of rapid notification of WHO that was made back in 2023, saying that targeted sequencing could be used in clinical practice and it could be beneficial from the standpoint of investments, and it could be effective from prescribing the targeted therapy. So how did we come up with the project? It was a long-term project that was hugely supported by WHO and a different number of non-commercial organizations, and the objective was to demonstrate the efficiency of targeted sequencing in terms of diagnosing multi-drug resistance products, and to make sure that it is accessible to the countries of middle and low income, which is really important. And actually those forms prevail in those countries. So we do realize that the major problem of multi-drug resistant forms of tuberculosis is that there's no funding in terms of treatment and keeping those patients in a good shape. So we picked 3 projects in India, South Africa and Georgia, and there were pilot stages of the projects conducted there. So the objectives of the projects were to have full-fledged genome sequencing, just like our colleague has mentioned about earlier, and we would identify the potential mutations that would be considerable from the clinical practice standpoint, and so that we could have the database and knowledge in regards to how to treat those mutations and to have a catalog developed accordingly. Furthermore, the phase of the project was aimed at introducing those innovations into the clinical practice and having it tried on in terms of efficiency and having full-fledged inpatient clinical trials. We do realize that it could work in theory, however we had to prove the clinical efficiency, which is really important, and it has to be cost-effective at the same time, and it has to be expedient from the funding standpoint. We have to make sure that those solutions are more cost-effective and less budgetary intensive, let's say, for the healthcare system. And this matters when it comes to large number of patients, for instance. And at this very stage, we decided to develop a couple of systems, as well as having the criteria designed on how to assess the efficiency of those systems. And the criteria would be comprised of the following lists of characteristics. So master profile sequencing. So any technology of sequencing of the new generation is to have a solution that is going to be based on the clinical sample. It has to be salina, or it has to be the tissue, and at the same time there has to be the full-fledged solution to include the preparation, which is to not have the pathogens and allele of resistance. It has to demonstrate to what products the resistance has been registered for, and the criteria of clinical efficiency were assessed at the level of 93%, and based on what I've just said and based on the criteria, we have actually the 3 products that could be good to go for the multi-drug resistance studies. And those products were provided by the company Genome Screenplates, and Oxford provided Nano-TB kit, and Shantin Biotech, Chinese company, actually presented NGS panels. And all of these panels would have different types of validated alleles of sustainability, and actually that was put all together. It's not only the drug resistance, and aluminum would not be aluminum if we don't have the MOU with GeneScreen, and if we would not develop the joint solution so that we could introduce it into the clinical practice, and in order to enable us enhancing the access to patients who live in our country. So to conclude, I have 7 minutes before I finish my delivery. So we have developed different training courses based on this large project, and I think that it is worth mentioning about getting connected, and you could use those links. You could ask for a PDF copy or just simply use the link, and there's a great deal of information contained there. It is the bioinformatics is described very, very detailedly, and the attitudes of patients as well as their behavior has been thoroughly described. So, plus, in addition to what I've just said, there's a free of charge course, the webinars and the control assignments that you may have as well. You could actually have bioinformatics based scenarios, and this is the product of the project. Now let us talk about Illumina and GeneScreen becoming the partners. I won't comment more on this slide, but the only message that I want to make is that we don't pay enough attention actually to diagnostics of infectious diseases, to include TB. And Illumina, for instance, pays more attention to cancerous diseases, to orphan diseases. However, as Pavel was mentioning in his presentation, infectious diseases is #1 reason of death, to include TB. And my mission actually is to compensate this gap. So consequently, GeneScreen is a small startup established in France. So for some reason, there's a great deal of interest to TB studies. Perhaps that's it is related to the fact that Institute of Dr. Pasteur is located there. I don't know. So they developed this platform. So very briefly, let me get you exposed to the peculiarities. So detection of resistance to 15 and TB drugs in 24 and 48 hours, and detection of minority variants that could potentially confer the hetero resistance down to 1 to 3%, and culture-free or only cultured isolates, and which is really important is that MTBO strain typing and identification more than 100 non-tuberculosis mycobacteria. So you purchase the platform and you get an access to software, providing you with the clinical reports. So we have presented the reports in two types. As a GenoScreen partner, we are exclusive distributor of the product. We distribute this panel, and it is available in two options actually. So those who haven't used the system before, we have different solutions, and solutions could be more or less expensive accordingly. And what we offer actually is that this software package good to go for the clinical package, which is not related to sequencing hub. Not all of us have an access to. You have to pay for the access and so on. It's not convenient, and our state enterprises can't announce the bidding, so the price of the analysis is included into the price of the kit. So the presence or lack of resistance is identified, and actually typing of mycobacteria is available. So I'm leaving 2 minutes to the next presenter, and I haven't included this slide into my presentation so far. However, my colleagues from Kyrgyzstan, Ms. Gulmira Kalmambekova, actually agreed so that I could mention about her achievements, which was working with 750 patients with multi-drug resistance, and she plans to publish the results. So in fact, we could ask for reference, and so far we don't have the works with the clinical data, as the project on testing was over a month ago. Thank you so much. Thank you. Could you please send us the link? I have a question for you. A very practical one. So how much could you load actually in order to... Well, unfortunately, I didn't include this slide. Should we use my ISAC? Okay, let's start with ISAC, right? We could load 13 samples. If it is MySec, if we take microkit, so it's 14 samples, 3 control, and 11 patients. Should we work with version 2, 150, then it's 24 samples, and if it is A3, 300 cycles, then it is 48 samples. So in Kyrgyzstan, the flow is not large, so they load 14 samples per cartridge. For them, it's more cost effective. If the flow of patients is larger, we could load either 24 or 48. We could actually use minisec, but the flexibility is less. There's middle put, high put, and in case of high put, we could do the 48. I have a small comment. Seven years ago or so, when we started in Russia sequencing very actively, we would work with full genome, and at the same time we would write about virulence and pathogenity of genes, and they would tell us, why do you need to have targeted sequencing? I'm glad that we are back to the practice. So should you know from a target standpoint where to put genes that helps. Yes, so that allows you to spend less money. So let's do the targeted sequencing. Yes, in fact, full genome is a more expensive method. So so there could be some answers. Thank you. Now let us greet next presenter. So, Ms. Galiyeva, please, the floor is yours. Good afternoon, participants to the conference. I just wanted to congratulate National Center for Biotechnology with such a prominent date and wish you to have more Nobel Prize winners in your organization. So allow me please to present my report, Application of Biomedical Technologies in the Treatment of Tuberculosis. Since I'm a chemist, we deal with synthesis of nanoparticles, optimization and increase of effectiveness of medicine. So I'm not going to be talking too much about prevalence of tuberculosis in the world. My colleagues told you that tuberculosis is killer number one amongst infectious diseases, and this problem still remains until now. Besides the fact that there's resistance and besides the fact that tuberculosis exists, there's multi-drug resistance predominantly to medicines, isoniazid and rifampicin. So in order to find a solution, lately, in the most recent decades, the researchers started using drug delivery systems. The systems that allow us to deliver the drug that to maximize therapeutic effect thanks to transportation and targeted delivery of the drug. So there are different systems of drug delivery. So it's those existing in nanoparticles, micelles, nanotubes, and today I would like to brief you about the application of nanoparticles to transport anti-TB drugs. Since nanoparticles have a couple of benefits, such as small size, they are more accurate, they have less side effects, and they have increased bioavailability, and it could be transported either orally or in inhalation. And nanoparticles actually do have different sizes in order to have nanoparticles delivering the drug into the alveolar macrophages. There has to be certain size of nanoparticles. The synthetic and non-synthetic polymers in order to produce nanoparticles, and you could see information in regards to synthetic polymers like polylactic, subglycolate, polyethylene glycol, and those nanoparticles get produced by simple and dual emulsion, nanopretation, and so on. And in terms of the natural polymers, we could name albumin, hitazan, and in order to have nanoparticles using the natural materials, we use chemical, physical methods as well as self-collection method. And this slide depicts information in regards to our developments with the use of synthetic and natural products. Isoniazid was used, rifampicin, combination of some drugs was used as well, and the results were incorporated. So one of the perspective polymers, natural polymers, is albumin, as it has great functional groups, and thanks to albumin we could actually dissolve the site and have the drug loaded. Actually, there is such a thing. A special characteristics of albumin, you need to stabilize it beforehand, before the delivery. So in order to have it done, we use a synthetic agent, which is glutardegide, and it has certain toxicity since it is synthetic. And in our method, we suggest using just natural polymers like L-cysteine, for instance. And this scheme demonstrates a drug immobilized albumin nanoparticles. So actually we start relaxation of the structure of albumin, so the hydrogen linkage, so by doing so albumin starts dissolving, so and that was achieved thanks to urea, and then we add some ethanol, and after that the dissolution gets started and we get nanoparticles. So in order to stabilize nanoparticles, we add L-cysteine. So actually we refresh SS bridges, and by doing so we have cross-linkage of nanoparticles. And this is done thanks to absorption or cross-linkage. So over here you could see synthesis of getting nanoparticles, and then the nanoparticles get characterized separately. So we identify the morphology, the size, the dispersibility, and so on, and the charge. Since the product is in the nanoparticle, then there's prolonged thawing of the product, and this slide depicts two methods. So the first one is a flow cell, and the second is vertical front cell method. So those two graphs demonstrate almost identical results. So therefore, the rate of rifampicin, for instance, release rate is identified, so there's explosion-like dynamics, and then the release starts getting slowed down, and we receive prolongation. So after having kinetic studied, what we do is we study microbiological activity, and in this regard we used a method called strain AH37RV, using the nutrition environment, and we identified the concentration of rifampicin in albumin, and our concentration in rifampicin was identified as 20 milligram per liter. So in this concentration, actually, the growth of mycobacteria of TB was not identified. And we decided to have a closer look at so it's a long way to do before we start clinical trials. So we have a long path to pass. During the preclinical trials, I wanted to thank our team, the Ministry of Funding or Finance, for providing us a grant. So thank you so much. Thank you very much. Will you have any questions, please? I'm now approaching the audience. So the photographs, was that made in your university? No, the photos were made not in the university. We don't have an access to mycobacteria, to strains. The studies were made at the physiatry department in Karaganda anti-TB hospital. And there at that department we have the studies, and we conduct the studies, get the results, and then we receive the results as microphotographs. No, do you mean nanoparticles? So we have scanning electronic microscope in Karaganda University, and plus we had 10 in Nazarbayev University, and we also had nuclear Microsoft, which we have. Thank you. Thank you very much for your attention. The next report is going to be given by Ms. Kurbambetova Gulmira, National Center for Biotechnologies. Dear colleagues, allow me please to make my presentation of Fusobacterium nucleatum in biopsied tissues from colorectal cancer in Kazakhstani patients. So this report is the results of the pilot project, and we collected them for 2 years in a row. So colorectal cancer is the term which is typical for ileum and other the major intestines of so it is 10% of all cancerous diseases on an annual basis. We have more than 3,000 cases of colorectal cancer, which is number 3 position after lungs and breast cancer. In accordance with the WHO, in Asian countries, Kazakhstan is occupying position number 15. After such countries as Japan, China, Korea, and so on. On an annual basis, more than 13,000 people die of cancer in Kazakhstan, and in terms of structure of death, the colorectal cancer is number 3 after lungs and stomach cancer. And just recently, there's one more factor as Fusobacterium nucleatum. Previously, that bacteria would not be referred to the evidence of reasons of colorectal cancer, and for the last 15 years that bacteria could be considered as a risk for colorectal cancer. And we have listed all of the reasons and risk factors for colorectal cancer, to include Fusobacterium nucleatum. And this is a gram-negative anaerobic bacteria, and normally it is found in the mouth, and it is related to parodontitis. And actually, amongst the most recent discoveries for colorectal cancer, there is a linkage with that bacteria. So the mechanism of appearance of that bacteria has different ways swallowing the bacteria. Next, at the same time, in the stomach there has to be a low level of acidity, and plus that bacteria could get through the bloodstream, and should in the intestine be dysbiosis, then that bacteria grows in intestine, and it could actually appear in case of inflammatory diseases in intestine, such as Crohn's disease and so on. And this slide demonstrates pictures of cultures grown by us with Fusobacterium nucleatum, pure Fusobacterium nucleatum. So from the recent researches for the last 15 years, there was a breakthrough in terms of having this as an evidence, saying that it could lead to colorectal cancer. Previously it would be saying that it actually just prevails in the tissue. However, in the most recent article says that thanks to full-fledged sequencing, Fusobacterium nucleatum was acknowledged as the risk factor for colorectal cancer. So we wanted to have a look at the presence of that bacteria in tissues of colorectal cancer in our patients, and in order to do so we had to cultivate and have the pure culture and have a full genome sequencing, and then have quantity-based PCR, and then to have a look at the expression of cytokines, the immune response of the body for getting infected with that bacteria, and then to get analysis of all data. And the materials and methods that we used are demonstrated on the slide. So biomaterial would be given by the National Center for Oncology. We have MOU with, and the doctors are included into the team of researchers. They were providing us with surgical material, locuses of tissues from different localizations. It would be the biopsy of the tumor, and then the neighboring biopsy, as well as the remote tissue biopsy 10 centimeters away from the tumor. So we would have 3 biopsies from one patient, and during 2 years those tissues would be delivered to the lab, and we would start away culturing. Specifically for Fusobacterium nucleatum, we would add antibiotics in order to inhibit the bacteria, and then we would identify the grown-up cultures, and then we would justify it with 16-test RNA to justify the cultures. And then in parallel to biopsy, we would have the DNA and RNA, and from the pure culture we would also have DNA for full genome sequencing. And on this picture you could see digital drop PCR. We would have the digital PCR for expression of cytokines, and we would have the full genome sequencing as a result. So out of all existent studies, in average such methods, quality PCR, digital PCR, Fusobacterium nucleatum would be identified in 40 to 60%. In our case, we identified 37%. And thanks to the MAUDI, we identified 8%.
Отличаются по стадиям заболевания. То есть early stage – это у нас первая, вторая стадия рака, и третья, четвертая – это late stage. Они между собой заселены фузобактериями практически незначимо различимы, то есть одинаково. Но и значимые различия у нас идут по самим локализациям опухоли. Это, как я говорила, уже сама опухоль, соседняя и нормальная ткань. Там коэффициент P-вавы составляет 0,02. Но и здесь ассоциируется именно...
Here we associate the bacteria with the Streptococcus, which has been detected not so frequently. That is why the results are not statistically valued. Here we compare the associations with the clinical characteristics of the patients, which we got with the help of physicians, and compared the persistence of Bacterium nucleatum with clinical characteristics of the patients. On this table, you can find out that the statistically valued data is the distribution of the bacterial distribution within the tissue, depending on its location. Cytokines, and here you can find the results of two cytokines, B1 and B6, and depending on the localization, we can see the differences between normal tissue and tumor tissue, between two interleucins. In the upper part, you can find the results of the digital PCR on concentration, and lower the data on correlation with housekeeping genes. And here you can find a correlation of interleucin with
Our project. Thank you so much. I have exhausted my time. We use housekeeping gene as we use the genome DNA. We would have it replaced, break it through to the strain, right? Yes. Yes. Yes, because the strain differently are different. That we haven't just presented them in this material. And the second question. All right. So in your publication, did you have any publications? Yes, we do on full genome sequencing, and the one publication is being considered on detection of bacteria. You made a correlation in regards to bacteria impact to expression of cytokines until level 6, right? So is it in comparison with the mainstream in terms of literature review and the mechanism that is typical for bacteria distribution? Yes. So as far as I'm concerned, on the contrary, the mechanism works all the way down, all the way around. So it's pro-inflammatory. Yes. So the host system, right? Yes, they've been actively generated while in getting infected. However, the bacteria may inhibit reproduction of those bacteria. So actually, what is the mechanism, and why is it contributing to the development? So in our results, the generation of cytokine is active, and they are differentiated, and those cytokines make the active inflammatory environment, and they provide conditions for viability of bacteria, so the tumor gets progressed because Fusobacterium, but in the literature it says just the same. Thank you. Thank you for your questions. Thank you.
Следующий докладчик.
The next presenter is Associated Professor of Astana Medical University, Ms. Burkitbayeva. I would like to greet all of the participants to the conference and the moderators of today's session. Allow me, please, to congratulate National Center for Biotechnology and the organizers of this conference with the anniversary, and on behalf of Astana Medical University and on behalf of the Clinical Pharmacology Department, let me do so. So my topic is personalized target treatment with biological products. And the relevance is that there are no diseases that could be named simple or complex. You know, for every single person, it's any disease has impact in terms of the quality of life. Unfortunately, the healthcare treatment is purposed to treat the disease but not the patient. And we forget taking into account the started, you know, showing it and so on. And now, unlike antibodies, is one of the ways you could get biological product. Getting monoclonal antibody is a complex, multi-staged process that takes several stages, starting with immunization of animals, and dependent on that, the medical products do get differ. And so this is chemo CMAP, humanized CMAP, and OMAP. So it's really important, actually, to decrease immunogenicity. So the biological products actually do have different effects. We may separate them into the monoclonal related to certain cells or proteins, cytokines, for instance, who may speed up or slow down biological factors, hormones or chemical substances that may be providing stimulation, and biological enzymes and immune biological modulators that have impact upon the immune system. In accordance with the data of the researchers under the U.S. Congress, top 10 most frequently used biological products are comprised of the list of 10 products that are used to treat such complex autoimmune diseases as what is the difference between biosimilar and bioproducts? You know, biosimilar is the same product that is equal to registered biological products in terms of the active substance. They have no differences, but at the same time, we can't say for sure that the difference with the generics demonstrate the biovalence and bioequivalency of the medical product is important. So biological or biosimilars have to be proven actually as the ones supported by pharmacological and genetical studies. You know, we can't sometimes pharmacological and there were 4 non-patent for use. It has to do with the autoimmune diseases, cancerous diseases, and in accordance with the instruction, the same biological product might have indications for different groups of patients. Thank you so much. The next presentation is going to be made by our guest from Belarus, Koneva Olga Mikhailovna. Please, the floor is yours. So good afternoon. I wanted to greet all of you. I represent Belarus State University and Biotechnology Lab, and perhaps yesterday you participated through the section in biotech for agriculture, and our chief made his presentation, and we work on having the recombinant proteins. And yesterday we talked about the agricultural application. Now we would like to talk to you bacterial glycosidase genes as a stage in creating molecular tools for targeted modification of antigenic determinants. Actually, those cells participate in different stages of synthesis, and actually they may get to the immune response. So should the antigen and antibody conflict, then this may lead to rejection. Actually, in 2024, there is the existence of 45 systems of antigens acknowledged, and in the clinics we would test just the two compatibilities. So the first was AB0 and RH3 system. So based on those AB systems, there are 4 groups of blood. So it's necessary to mention that individuals who have a certain antigen, there's not going to be antibodies to that. However, there's going to be antibodies to the missing antigen. So why do we have to control that? Because this could lead to transfusional reactions, should there be the blood transfusion, and as I said to you before, to rejection of tissue in case of transplantation. So the picture depicts the AB0 example, antigen determinants. So demonstrate the scheme. So the polysugar leftovers, so say, let's say cells are depicted on the screen. So antigen A and antigen B are depicted on the screen. So the product of gene expression is not the gene determinants, but whatever leads to linkages in oligosaccharides distances. So actually in each and every scenario you could see how the situation could look like and how it could be developed. So actually, why are we talking about the structural peculiarities? Is that the structural peculiarity makes it possible for us to have the reversed convergency, and this is going to be contributing to the fact that, for instance, the first blood group is deemed universal. So this approach actually has been used largely to get donor red blood cells. This was something that would be, started back in 80s of the last century, and then they would look at the universal O red blood cells, and there were not optimal conditions of pH, temperature for the enzymes, and they had to have, large numbers of, and anyway, this led to a great interest in studies, and one of the options was to study the microbiota of a human being, right? So why is it so? In epithelium tissue, we do have glycans, and its structure actually is the structure of glycans is really similar to the antigen determinants. And plus, on top of what I just said, the microbiotes of the intestine could use those glycans as the source of carbon. And actually, this leads to potential productive glycosidase. So in physical conditions, they would be, effectively splitting those oligosaccharide pieces of lactose and form the universal H antigen. And during functional metagenome analysis with the use of high sensitive studies, there were two enzymes that were identified, and we actually started looking at those proteins in Escherichia coli system. So why those enzymes are interesting? And so it actually colonizes the first stage, and then as a result we get the structure of the universal H antigen, and we have a release so those enzymes actually work, and they're pretty comfortable, in terms of transportation, and it has the physiological pH levels and comprise an interest. And actually, in terms of nucleotide sequence, those two enzymes were actually cloned, and based on those hybrid plasmids, we received the strains of Escherichia coli, as well as the sizes that are actually equal to the sizes. And over here I have described the characteristics for the second enzyme. So actually, the mutations were acknowledged by the sequencing. And the determinants, is really interesting to optimize. We have optimized the expression conditions, and we use the so 16 and 18 degrees were picked during 16 hours. The results actually allowed us to have a good strategy in order to have a good purification of protein. I tried to be brief, as we are behind the schedule, and I can respond to all of the questions during the lunch. Thank you so much for your report. So, do you have any questions?
В общем так, я хотела спросить, в дальнейшем, как вы планируете использовать?
I wanted to ask you, how are you planning to use your enzyme? So, in fact, this is the topic of our study was offered by our Institute of Transfusional and blood transfusion, and they want to have a special machine allowing them to pump the fluid that would have the bacterial glucosidase in order to use it for transplantation later on. And the intention is to have it. So we do the molecular part, and what they do is that they just deliver. So you use a lot of toxins, want your patients suffer. So
do plan to use the strategies for purifications of your product because you have the enzyme which is used from glycol will contact with human organs. You need to take a closer look at some set of indicators where we use the products, and we use a specific test which identifies the operative genes which could lead to these harmful consequences. This stage is a part of our work. We try to reach the maximum level of purification. We cannot get the super purified actually, but we tend to use the most purified as possible. That is why we consider the use of certain procedures. Will you do this within your lab, or you will send the developed approach to someone else? We'll create the ready-use package with code of use and so on. We are a governmental university. We do not require the ready-done platform for production. Thanks. In fact, they are the Science Academy, and they run under the ministry. Probably have something like, and so on. I have the following question. Does the team plans to run research on this? The specific intense gut microbiome bacteriosis, which interacts with glycosidase and targeted on intestine first of all, on the first stage, and what you need to identify on the second stage, which carries the genes, and is it possible for it to make a synthesis in such an amount? Thanks for the question. I have the clarification. Do you mean that we plan to search for other type of glycosidase? Yes, the other types, and for which particular purposes in terms of intestine function. With the help of antigenome analysis, it seems like we plan to do so, but now we are more focused on glycosidase bacteria within the particular project we have. But when we will have a request, we are ready for judo. We are open for the cooperation. Thank you. The next speaker is the member of Biotechnology Center, Birgen Belshahar. You will have 10 minutes for your presentation. Please consider the time. The mic is off. Good afternoon, dear participants of the conference. I would like to create the sense of a jubilee. Now we start to proceed. You know that is a type of biopolymers of life origin, including polysaccharides and polypeptides. For the first group, we mean ketosine, hyaluronic acid, and a range of polypeptides, including albumin. Within our work, we use four famous polymers: gel, hyaluronic acid, ketosine, and androgen sulfate. The key method is based on two pillars: on physical and chemical built polymers. We used both of them with the help of cryopolymerization. That is a process which occurs under the low temperatures, lower than solution. Firstly, we use the freeze dissolvent solution, and the second one runs in the lower temperature microphase and the initiating process. We use, as I mentioned, we use 4 polymers, and we got 2 different cryogels. First one is gel CH. We dissolved gel in certain ratio, 1:1, in the acid base, then the protonides in the base solution. For the ketosane in the solution, we added the hydrogen sulfate for getting the complex and getting the interaction of sulfate within the complex. Further, we used the syringe for transporting the gel and for the freezing within the 24 hours. Then we purify with the help of EDC sewing agent, actually. That was the main sewing agent we used. Within the 24 hours, we kept the gel in the normal solution. Then it was been purified to separate it from the alcohol, and then stabilized it. We used the cryogel, cytosine, and creatine sulfate. The key indicators have been the components of the gel and all the characteristics you can find out here been verified with the use of methods of interaction of medical solution with the help of substitution of the fluid and absolute alcohol. You can take a close look at the table and found out where we use hydrogen sulfate 100, 150. We have 2 samples which gained the best results. In terms of density, it's been much higher than in other samples. The components of the gels been identified with the use of microscopy methods. As the main factor, we've seen the growth rate of gel, and there has been a need to use the polymers as biomaterials. And when we see biogradation, they should be separated into the elementary oligomers with no disruptions. And the lower degradation we have, the better features polymers have for the further use. You can take a look at the first graph, upper graph, with the base on 50 and 100. They result in different characteristics depending on their density, actually. The more polymer mass we have, the more sorbent features we have in polygels. Apart of that, we researched the morphology of the cryogels, and all the cryogels depict the following structure, open typed. And the further research of these polymers has been stopped. The major cryogel has been in the base 100, as you've seen. It has the diameter of 230 nanometers. Further, we explored the cytotoxicity effect on the lipid tissue of rats. We used the stem cells, the liquid solutions, and we discussed their impacts on the live cell. As you can see from the data we received, the better result depicted from the sulfate 100, and further after the dissolving the hydrogen sulfate 100 and more get the following results. They have a higher, they are less toxic in comparison with the alternative ones, and they could be used as the biological matrices. The use of same approach, we've got the other solution. As a result of previous research, we had a ratio between the gel and creatine sulfate 4:1 ratio. We used the CBS solution.
So we created sulfate, we worked with an nitron acid, and then we would dissolve it, and the principle was the same, so we would just frozen it up, and then we would have it melted, cleaned up, purified, and we would add cross agent in the end. So the studies were conducted in order to have the first fraction and the porosity of the polymer. So the higher the fraction is, there's less salt and there's less waste. And the porosity plays a great deal of role. Actually, we use stem cells, and they do have a certain size and certain weight. So they got to be attached to the net so that they can feel themselves comfortably. We looked at the reaction to the gel fraction of polymer. So the identification was made in synthesized computerscopy. And we also had the studies in terms of biodegradation. As I have demonstrated earlier, in the studies, the polymer itself, is having from the degradation standpoint, the results have demonstrated the more fluctuations we have in terms of the results. As there's a transition in quantities, the largest biodegradation is typical for cryogels in contact of hyaluronic acid with 10 milligram. And next is a synthesis of cryogels, as well as the water. And in case of this flow solvent, then they've demonstrated the result in terms of viability. Thank you so much for your attention. Thank you. So next presenter is Ms. Aygul Sutimbekova. She's going to be talking to us about the dynamic studies. So,
жолындағы алғашқы мектебім ол ұлттық биотехнология орталығы. Вячеслав Борисович менің алғашқы ұстазым болып табылады. Ұлттық биотехнология орталығынан ғылым жолына толағай табыстар тілеймін. Бүгінгі менің баяндамамның тақырыбы көп бейінді ауруханада ацинотобактер баумани қоздырғышының динамикалық деңгейін анықтау және де оның антибиотикке тұрақтылық сезімін анықтау. Госпитальді штамдардың ішінде ацинотобактер баумани қоздырғышы қазіргі таңда соңғы жылдары көптеп кездесіп жатыр, және иммундық жүйесі төмендеген науқастардың арасында асқынулар мен өлім жітімге әкелу себебіне байланысты бұл ацинотобактер баумани қоздырғышы өзекті мәселелердің бірі болып табылады. Жалпы дүниежүзілік денсаулық сақтау ұйымының антибиотикке тұрақтылық антибиотикке тұрақты микроорганизмдердің 7 сатысына дабыл қағуда. Соның бірегейі ацетобактер баумани қоздырғышы. Біздің зерттеудің мақсатында жалпы балалар кардиохирургиясы және де ересектердегі қарқынды терапия, реанимация, анестезиология бөліміндегі науқастардан алынған материалдардың динамикасын жасау және де антибиотика граммасының реттілігін анықтау жұмысы атқарылды. Зерттеу базасы ретінде Бисенова Михайловнамен бірге Астана қаласының ұлттық ғылыми медициналық орталығынан материалдар алынып зерттелуде. Зерттелетін материал ретінде реанимация және де балалар кардиохирургиясына түскен науқастардың биоматериалдары алынды. Оның ішінде ең жиі кездесетін плевра сұйықтықтары, қан және қақырық, жарақаттан кейінгі жұғынды трахеобронхиалды ағаш бөлімінен бөлімділер алынды. Және таза дақылды бөліп алу үшін Витек эко компакт анализаторы қолданды. Ол жерде идентификация және де антибиотикке тұрақтылық реттілігін анықтадық. Жалпы зерттеудің нәтижесінде анестезиология, реанимация және де қарқынды терапия бөлімінде жалпы осы 2015-2023 жыл аралығында 202 ацетобактер баумани штаммы бөлініп алынды. Оның ең жоғарғы дәрежелері трахеобронхиалды ағаштан және де жоғарғы тыныс, төменгі тыныс жүйелерінен бөлінді. Ал балалар кардиохирургиясына келетін болсақ, ол жерде жалпы саны 427 ацетобактер баумани қоздырғышы бөлініп алынды. Ең жоғарғы көрсеткіш бізде жоғарғы тыныс мүшесінде анықталды. Бұл біздің ацетобактер қоздырғышының динамикалық дәрежесін бақылау, жылдар аралығындағы реттілігін, ересектер реанимация және анестезиология бөлімінде ең жоғарғы көрсеткішті 2015 жылғы көрсеткішті қарауымызға болады. Ал қазіргі таңда ересектер арасында микробтық пейзаждың өзгерісіне байланысты соңғы жылдары штамдардың санының төмендеуін айтып кетуімізге болады. Ал балалар кардиохирургиясында керісінше соңғы 2021-2022 жылдар аралығы осы зерттеудің ең үлкен пигіне жеткен пейзаждық қоздырғыш деп айтуымызға болады. Бұл балалар кардиохирургиясында өзекті мәселенің бірі болып табылады. Мына жерде динамикалық деңгейін бақылауымызға болады. Балалар кардиохирургиясында және де анестезиология реанимация бөліміндегі ересектердегі кездесетін ацетобактер қоздырғышының динамикасын қарауымызға болады. Балалар арасында соңғы жылдар аралығында осы қоздырғыштың штамдық деңгейі жоғары көрсеткіш көрсетуде. Сонымен қатар зерттеу үшін біздің микробиология лабораториямызда 7 антибиотик қолданылды. Оның ішінде мирапенем, имапенем және де ципрофлоксацин, левофлоксацинмен аяқтайтын топтар. Бұл жерде көрсеткіштер бойынша зерттеу нәтижесінде ең жоғарғы көрсеткішті левофлоксацин 93,3% антибиотикке тұрақтылықты көрсетті. Ол дегеніміз қарқынды терапиядағы антибиотикке тұрақтылықтың өте жоғарғы дәрежесі екендігін көрсетеді. Ары қарай балалар кардиохирургиясындағы нәтижесі салыстырмалы түрде ересектермен қарағанда қуантарлықтай. Алайда бұл жерде имапенемге 27,8, левофлоксацинге 27,3% деңгейде антибиотикке тұрақтылық байқалады. Қарасақ, ересектердің арасында қоздырғыштардың саны аз болғанымен антибиотикке тұрақтылық дәрежесі өте жоғарғы деңгейде. Ал балалар арасында саны көп болғанымен антибиотикке тұрақтылықтың деңгейінің төмендігін байқаймыз. Ол дегеніміз балалар арасында микробиологиялық бақылаудың жоғарғы деңгейде тұрғаны штамдық өзгерістерге антибиотиктердің сезімталдығы байқалады деген сөз. Қорыта айтқанда, жалпы 2015-2022 2023 жылдар аралығында жалпы бізде 629 штамм бөлініп алынды. Олардың 32,2% реанимация және қарқынды терапия бөлімінде, ал ересектерде 67,8% құрады. Сонымен динамикалық деңгейі бойынша балаларда соңғы жылдарда, әл ересектерде керісінше 2015 жылдар аралығында штамдардың көптеп кездескенін білеміз. Сонымен қатар антибиотикке төзімділіктің нәтижесінде карбапенем, фенолон, аминоглицидтерге антибиотиктер төзімділік көрсетті, ал колистинге төзімділік табылмады. Қорыта келе айтарымыз, ацетобактер баумани штамдарына қатысты табрамицин, амикотин антибиотикті терапияларды қолдануға, кешенді пайдалануға болады. Сонымен қатар, ацетобактер баумани қоздырғышының госпитальдік штамдарын алу мақсатында осы реанимация және балалар кардиохирургиясында микробиологиялық бақылауды үнемі жасау, ұтымды микробқа қарсы препараттар және де инфекциялық бақылау стандарттарын қатаң режимде ұстауды ұсынамыз. Назарларыңызға рахмет.
Рахмет.
Unfortunately, we don't have time for questions anymore. So let us have the last presentation in this section. Alия Sikhenova, National Center for Biotechnology, please.
I'm Alия Sikhenova, PhD, Senior Researcher of Stem Cell Laboratory. It's a great honor to me to participate in this conference dedicated to the 30th anniversary of this center, and also my gratitude to organizers of this conference. Let me present my small findings of our study, the therapeutic potential of preconditioned mesenchymal stem cells in atherosclerosis. There is content of my report. So what is atherosclerosis? Atherosclerosis is inflammatory chronic disease which is associated with the formation of lipid plaques and also with insufficient blood supply. The risk factors are associated with hypertension, diabetes mellitus, smoking, mental stress, and poor nutrition and lack of physical activity. Pathogenesis of atherosclerosis, according to the recent studies, is associated with the chronic inflammatory processes. It consisted of entrapping of oxalate production of integrin and selectins, transdifferentiation of monocytes, and plaque formation. The current therapy is presented with statins, antiplatelet drugs, and anticoagulants, and by coronary angioplasty. But the limitation, despite of advances of pharmacological drugs, there is still intolerance and resistance and toxicity at patients who suffer from atherosclerosis. And in our study, we are thinking about the alternative approach to treat atherosclerosis. And according to the recent studies, mesenchymal stem cell possess immunoregulatory and immunomodulatory properties and regenerative potential, which are mediated with cell-cell contact and paracrine mechanisms. And according to the recent studies, the mesenchymal stem cells showed atheroprotective, regenerative, and immunosuppressive properties in animal models of atherosclerosis. But we are thinking also about how to improve the immunomodulatory properties of mesenchymal stem cells. And we propose a preconditioning using the hypoxia and using the cytokines. Also, there are other ways, using of 3D spheroid and genetic modification. So mesenchymal stem cells are sensors and switches of inflammation, and they can balance polarization of monocytes and to M1 and M2 macrophages. So there is a plan of our study. We aim to evaluate the therapeutic efficacy of preconditioned mesenchymal stem cells in animal model of atherosclerosis. There is a design of our study. We do induction of atherosclerosis in upper mice by using atherogenic diet. So there were three groups: the control groups, the MSC group, and TNF MSC's group. And we used 2 million of cells, which were injected 2 times by 7-day interval. We used ELISA, flow cytometry, microscopy, and histology methods. So our results, we obtained mesenchymal stem cell, which have normal characteristics of cells, which we can see by CFU, multi-image differentiation, and expression of normal markers. The next one, also we tried to develop 3D spheroids, and we obtained 3D spheroids which totally contained of mesenchymal stem cells. The next one, that our cytokine preconditioned MSCs showed showed the secretion of immunoregulatory factors such as PGA2 and interleukin-6. The induction of atherosclerosis in upper mice were proved that after 60 days we see the formation of plaques in the aorta in the aorta that were isolated from upper mice. The next result is that TNF-alpha preconditioned MSC decreased the level of pro-inflammatory cytokine levels, namely the level of TNF-alpha by 2-fold and interferon gamma by 4-fold, and also increased the level of anti-inflammatory cytokines. Also, TNF-alpha preconditioned MSCs decreased the level of cholesterol and LDL slightly, and increased the level of HDL by 2.5-fold. The next result is that TNF-alpha preconditioned MSCs restore spleen weight, and the size were decreased by 1.6-fold, and the weights were decreased by 4-fold. The next results are showed that TNF-alpha preconditioned MSCs reduced the size of the plaque by 78%. And conclusion. So we obtained MSCs that had normal characteristics of mesenchymal stem cells. The cytokine preconditioning improved the immunoregulatory and anti-inflammatory properties of MSCs. We showed that TNF-alpha preconditioning have a significant immunoregulatory and anti-atherogenic effect in animal model of atherosclerosis. So the data can be, our obtained data can be prerequisites for the new therapeutic methods based on TNF-alpha preconditioning. The results of this study were published in two papers, in research and review papers, in impact factor journals. Thank you for your attention. Назарларыңызға рахмет. I thank to everyone who contributed to this study. To my great duties to Professor Ogai, who was my supervisor, to Professor Vaidera, who also was my scientific supervisor, also to my university, and also to stem cell laboratory of this National Center for Biotechnology. Thank you for your attention.
Спасибо. К сожалению,
Thank you very much. Unfortunately, we don't have time for questions. You may ask your questions during the lunch break. Now we're going to be having a lunch break, and there's going to be a small announcement to be made. So we're going to be getting together at 2:30 sharp for the solemn ceremony of providing the awards and certificates, and the winners prizing ceremony, so please don't hesitate to participate. Thank you so much for your contribution and participation, and enjoy your lunch.
Құрметті әріптестер,
Dear guests and dear participants, dear guests, so our scientific conference is about to end. So we would like to thank you for making it and participating. So the ability to get exposed to your experience and practices was a great deal of success for all of us. Dear colleagues and dear participants and guests, the conference has gone to its end, and we would like to express our sincere appreciation for each and every one of you for your contribution and participation. Your discussions and presentations and the exchange of experience made this conference absolutely unforgettable. And we have been exposed to a wide range of different innovations in terms of biotechnology. We had a chance to discuss different areas of interest. So within those days, we had over 100 presentations that has covered a wide spectrum of different studies in biotech. Now, allow me please to invite General Director of National Center for Biotechnologies, Professor Olga. The floor is yours. Good afternoon, dear guests and participants to the conference, dear friends. From the bottom of my heart, I would like to congratulate you with the successful completion of Astana Biotech 2024, devoted to the 30th anniversary of our center, National Center for Biotechnology. You know that Astana Biotech was one of the objectives in order to demonstrate the results of studies made by Kazakhstani and our foreign partners in agriculture, environmental protection, food processing, and biological safety. Within the framework of the conference, we also conducted a symposium devoted to regional forum in Kazakhstan, biodiversity, and thus we have incorporated 320 scientists from all around the world who represented 30-plus scientific organizations and universities from Kazakhstan and other countries, such as Germany, Hungary, Netherlands, China, the United States, Pakistan, Russia, Belarus, Uzbekistan, and Kazakhstan. Within the framework of the conference, we had plenary and section-based discussions where we had oral presentations in 4 areas, and the conference was incorporated of 40 presentations in biomedicine, genomics, regenerative medicine, agricultural and technologies, environmental protection-related technologies, and biosafety. And the presentations were made by prominent scientists such as Rafael Sohn, New York University of Abu Dhabi, Aman Ali, Russian R&D Institute for Medical Products and Devices, Jozef Shaki, Teki University, Hungary, Mr. Makrausov, St. Petersburg, R&D Institute for Microbiology, named after Pasteur, Goncharov Institute of Biotechnology and Molecular Bioengineering, Danilenko Valery Nikolaevich, who represents the Institute of Common Genetics, Filipenko, Institute of Chemical, Biological and Experimental Medicine, Novosibirsk, Rinbedov, Kenya's Russian Technology University, Gazovaya Anna Alexandrovna, St. Petersburg, R&D Institute of Epidemiology, Kirichenko Anastasia Sergeyevna, R&D named after Almazov, Valeria Buzuk, representing Bruegger Daltonics, Novikov, Omsk State University, Alakpash, Punjab University from Pakistan, because Patagoyev Maxim, Belarus State University, Sherpakova Larissa, Russian Institute of Pathologists, Mustafina, Botanic Garden, named after Rusanov, Tashkent. Javahi, Vitaly Georgievich, Russian R&D Institute of Pathology, Statyuk, Natalia Vladimirovna, Russian R&D Institute of Pathology and Institute of Microbiology. Evgenia Nikorova, Russian Institute of Plants, Zorov Ivan Nikitich, Experimental Association, and Ryshkova Alexander Mikhailna, Experimental Foundations of Microbiology, Institute of Microbiology, named on the Babach Federal R&D Center for Gardening, Moscow. And the conference was given the floor, has been used in order to give the floor to the most prominent scientific institutions from Kazakhstan. Those Sarbasov, Nazarbayev, Uzbekov, Institute of Microbiology, Besimey, Ahmed Gatshi, Eurasian National University, Sadanov, Aman Turbanovich, Institute of Microbiology and Virology, and Ishmaratova, Suminbaev, Adral, Bugintaev Sirik, Argenberik, Almerikova Shirin, Institute of Biology and from the same organization, Mr. Белозеров. So Terletskaya, Nina Vladimirovna, Institute of Genetics and Biology, Kazakh National Agrarian University, Japarkulova, Kazakh National Medical University, Zhunusov, so Director of the Institute of Genetics and Physiology, Burkitbaeva, Medical University Astana, Eurasian National University, and R&D of Genetics under the Kazakh Academy of Science. Alihan Bukikhan University, R&D Institute for Biological Safety. Kazakh National Medical University under Asfendiarov and Zhigalov, representing branch office of our center in Almaty. So we have listened to presentations in a wide range of areas to include So the moderators of the conference were Abdul Sarbasov, Danilenko, Kirilenko, Turspekov, Kalendar, Sagaliev, Mamadalik, Bukintaev, and the moderators were the leading scientists of our center. So we also have arranged the exhibition with the participation of our sponsors, those who produce lab equipment, to be precise, Ile, Tsalma, Baking Biomedical, Data Lab, Jekernau, ATT Bio, Optonic, Conveno, the same Kazakhstan, Algimet, Bioconus Plus, Carl Zeiss, Kazakhstan, Medialand, Tech Prime, Ines, a company called Tetis, Microsolutions, and a National Center for Biotechnology with a branch office in Stepnogorsk. So thus, we hope that this event, Astana Biotech, is going to be actually giving a new impetus to discussions of biotech and having the more strengthen ties amongst the scientists from different countries, and you will get exposed to new technologies. And by doing so, you have contributed to the development of the market of biotechnology and introduction of new innovations in real industrial sectors of our countries. And to complete, summing up, I would like to give the floor to our distinguished officer. So he used to be deputy director responsible for science. So he was one of the pioneers and professor, doctor of veterinary science, a great professional in veterinary and agricultural technology. Kasim Mukanov, please, the floor is yours. Thank you. Dear colleagues and dear participants to the conference, since yesterday we've been celebrating the 30th anniversary of National Center for Biotechnology, and we're coming to the closure of the solemn ceremony. So I would like to greet those who have joined us, and I would like to say thank you to those who have made it from abroad. And yes, you've been sharing very warm words, so let this all be true, and I would like to wish you success. And let's get together in the future, and let's discuss very important issues of mutual interest. So, dear participants to the conference, dear guests, and since yesterday we had a great dynamics and we had a very nice atmosphere. You know, it was not just a creative atmosphere. You know, we met with those whom we haven't met for years, and we've listened to really interesting presentations, and Mr. Ogai listed those who made their speeches. I'm not going to be repeating the names. It's a great thing when people get together and share the achievements and progress. And I would like to thank our guests from abroad, from Russia, Uzbekistan, Pakistan, Hungary, Germany, and other countries, who have joined us and who have participated to this conference and who have expressed their warm words. I just wanted to say a couple of words and going back to the history, I won't take too much of your time. I joined biotech industry in 1985 on September the 1st, as I remember it perfectly well. We received a large delegation chaired by General Secretary of Communist Party, Mr. Gorbachev, and we also had a presentation of President of Soviet Academy of Science, Mr. Chivnikov, and Mr. Chivnikov making a speech in Tselenograd Agricultural Institute actually was briefing us about the biotech as the new wave in science, and Caroline Milstein opened the technology of having monoclonal antibodies, and they were those who became the winners of the Nobel Prize. And Mr. Sagadiev made a speech, and he asked, what if we open one of the centers here in Tselenograd? And for some of those who were present, were wondering, it was a regional center. However, it became true, and 1 week later we traveled over to Moscow, and we came to Moscow, and Rita Dutch asked why so fast, and we said, well, we have to, start at once. So since then, since 1986, there were 13 people who were accommodated in Moscow, nearby Moscow, and we have completed internships there. So in 1987, on January the 7th, the decree was issued, and in accordance with this decree, 14 biotechnology centers were created in the Soviet Union. So 2 of them were in Kazakhstan, one in Almaty and one in Tselenograd. So why am I using this name Tselenograd? So Tselenograd Agricultural Institute was the basis of that center, and we've been given premises and so on. So in 1993, the center in Stepnogorsk was established, and until 2001, the National Center for Biotechnology, the HQ, was located in Stepnogorsk. So in 2001, so those were the circumstances. When Mr. Abdikanov chaired the center, actually we contacted Mr. Bekturganov, the minister, so the government supported the idea. So Tselenograd Agricultural Institute provided 4-storied building with the personnel members, with the furnitures, the chairs and tables. That was given to the balance of the National Center for Biotechnology. So the National Center moved from Stepnogorsk to Tselenograd, and actually since then we started working in biotechnology, and we have completed the internships and so on. So after that, when we moved to Tselenograd, then the new era started. There was a great decree as of 2006, the 3rd of May, 329 was the number of the decree. So that decree actually approved such a great R&D program with vast funding, and implementation of the program engaged 845 scientists from 30 scientific organizations, and 9 organizations and universities were those from Israel, US, Japan, and France, Russia. So we would not just make the works, we've been gone through the internships. And Mr. Hasenov, I'm not going to be listing, So we had 1 to 2 year internship. So, and I have to say that Tselenograd Agricultural Institute played a very important role in terms of establishment of our center. So the center is the product of everybody's effort, and now our center is not the center providing the abilities for in terms of fundamental studies. We have master's degree, doctorate degree programs, we have interdisciplinary studies, and we partner with the leading universities from all around the world and Kazakhstan. I'm not going to be listing our partners, so therefore I'm so glad to have our white-beard wise men coming to this conference. Mr. Mamadaliyev, who worked with us since day number 1. We used to be colleagues before, so actually I think that we have to remember these things. And we have to keep following the traditions. I'm so glad to host a lot of guests, and we listened to really interesting presentations, and this is going to be a stimulus for all of us to give, and it will give another impetus to the development of science, so that we could be partnering with you all. I'd like to say thank you for organizing this event. Let us give a round of applause to the young scientists. They spent 24/7 Department of Science, Administration. Thank you so much. And thank you full-heartedly to all of our guests. All the best and success in all of your endeavors. And see you soon during the 6th conference. Thank you and see you later. So now we are going to be mentioning those who have won the diplomas, and let me invite Mr. Ogaev to the scene. The next stage of the ceremony of closure is going to be awarding the best presenters of poster presentations. So, Mr. Ogaev, please, you are given the floor. So, dear guests and dear participants to the conference, so here comes the most prominent event, which is the awarding ceremony for the best exhibition and the best oral and written presentation. I'm so pleased to give you awards with the small tokens of appreciation and diploma. So the best stand presentation in biomed technology, Novikov Dmitry Georgievich is getting the prize. The next award is given to the best oral presentation in innovation biotechnologies for agriculture, Zorov Ivan. And the next award is given to the best presentation in biosafety, Mr. Stapchuk. Next, so biotechnology for ecology, food and processing industries, Ms. Rashkova. Now we're going to be shifting over to the nomination for the best poster presentation, and this award is given to Bektayev Rahimbek. So, National Lab Astana, Nazarbayev University, one of the best scientific organizations in the country. And a nomination, the best exhibition stand, the best company who has arranged its participation participants to the exhibition Micro Solutions, please. As you know, within the framework of our conference, we had symposium devoted to flora of Kazakhstan, and we had participants from all over the world, Uzbekistan, Kazakhstan, and I wanted to give the awards and diploma. Ms. Firuza Mustafina, please. So diploma for the second place is given to Tatiana Vdovina. And this is the third place winner. Muldir Nurmagomedova, please. And there's one more award, which is a thank you letter by Balnur Kali for the best post presentation. Yes. So dear participants to the conference, I just wanted to make a small announcement. So all of the photo materials are going to be placed on the website of our conference. There's going to be a link to Google Doc, and you can download all of the pictures as a result of the conference. Thank you. So with this, dear guests, dear colleagues, allow us to solemnly finish Astana Biotech 2024. We would like to thank our technical personnel members. So thank you so much for your professionalism, and thanks to your efforts, we could have this event arranged at the highest level. So special thanks to the organizers, volunteers, and technical services who made it possible at such a high level. So without your endeavors, this conference could not be possible. So dear participants, we hope that the knowledge that you have possessed during this conference is going to be used in your practice. So let us meet each other during such prominent events. So dear participants, we have no doubt that the knowledge that you have gained during this conference is going to be fostering the science and practice. Once again, thank you all for your participation, being full of energy and passion for science. So all enjoy success in all of your endeavors, and see you next time. Goodbye. With this, the official part of the conference is over. Let's give a round of applause to each other. Thank you.